With more than 6,000 small and micro-cap companies listed, if you're looking for the next Apple at the earliest stage, then ChannelCheck truly is the orchard. The listed companies support ChannelCheck, so it's free for you, the potential investor, to gain access to institutional quality research from FINRA licensed analysts, advanced market data, industry reports, news, and a growing catalog of videos and webcasts. Make sure to subscribe to our YouTube channel and register for ChannelCheck so you're always up to date on what's going on at the small and micro-cap data place. Welcome to this special edition of the C-Suite series presented by ChannelCheck and Noble Capital Markets. Noble is an SEC-registered FINRA-licensed broker-dealer and the source of the equity research available on ChannelCheck. This investor forum at the World Stem Cell Summit presentation features ExoProther Medical. Following a brief overview presentation from co-founder and CEO Dr. Lana Volek, Noble Senior Research Analyst Robert LeBoyer will moderate a Q&A session. If you registered for this event through ChannelCheck.com, in the coming days you'll be given an opportunity to get your own questions answered by the company. With that, I am pleased to present Dr. Lana Volek.
Hello, I'm Lana Volek, co-founder and CEO of ExoProther Medical. And we're on a mission to bring to patients a breakthrough therapy targeting about 60% of human cancers. The name of the company, ExoProther, actually encodes our innovation, exosome protein therapeutic. We use exosomes, native nanoparticles produced by cells, to deliver wild-type p53 to tumor cells, translating a newly discovered local mechanism of anti-cancer defense into a systemic therapy. p53 is known as guardian of the genome due to its central role in cellular response to stress. In case of DNA damage, p53 either corrects the damage or triggers programmed cell death, apoptosis. When p53 is not active, DNA protection is lost, leading to cancer development. About 60% of human cancers are associated with mutation of the gene encoding p53 protein. p53 mutated cancers are our target. Our exosomes originate from unique tissues where cancer occurrence is extremely rare. These tissues are characterized by high content of wild-type p53 protein and complete absence of its ubiquitin ligase MDM2. In these immunoprivileged tissues, the main anti-tumor defense originates from neighboring cells. In case of damage to p53 encoding gene in some cells, these cells receive wild-type p53 from surrounding cells through exosome transport. This leads to restoration of anti-cancer function in the cells. We translate this newly discovered ancient local mechanism of anti-cancer defense into a novel systemic therapy. We harvest those native exosomes, purify and scale the process, and use them as delivery vehicle of the anti-cancer therapy. So we create a process that is parallel to the one that has emerged through 600 million years of evolution. Our therapeutic target is malignant cells with mutant or inactivated p53. The product is a unique complex containing native nanoparticles loaded with tumor suppressor factors, including wild-type p53. And the therapeutic effect is selective apoptosis of malignant cells and potential sensitization to conventional therapies. We target a range of high-impact indications based on the percent of p53 mutations and the current unmet need. These are GBM, the brain cancer, lung, bladder, colon, and ovarian cancers. What is important is that delivery by means of exosomes ensures flexible and multiple administration routes, such as inhalation in case of GBM or lung cancer, and it has been shown already that exosomes are able to penetrate BBB both with systemic administration and through nasal inhalation. There are other possible administration routes And in all cases, our therapy can be combined with existing therapies. On top of this list is GBM, where more than 90% of patients exhibit p53 mutation, and current 5-year survival rate is just 5%. So by targeting GBM, we have the potential to create huge impact on an important patient population where currently no cure exists, and pediatric patients comprise up to 15% of this population. Devastating side effects of conventional therapies are even more severe with targeting brain cancer patients. And there is the huge brain-blood barrier challenge for conventional therapies, while a potential for a non-invasive administration of exoprothor therapy. In terms of business potential, this is still a significant market, growing with CAGR of 7.5%. In addition, this indication has potential to receive orphan drug designation and other incentives by FDA for accelerated market access. And clinical researchers and the market are very accepting to trying novel GBM therapies. There exist multiple no-profit collaborative initiatives aimed at speeding up access to clinical trials for potential GBM therapies. Today, GBM patients are treated by conventional therapies, combining surgery, if it is possible, with radiochemotherapy. Patients suffer from devastating side effects, but still have very short life expectancy. GBM is known to be resistant to conventional therapies, actually due to the high percent of the p53 mutation, which compromises cell responses to DNA-damaging agents, such as radiation and chemotherapy. For the last decade, the only therapy approved for GBM indication was the Novocure device. Unfortunately, the treatment only slows disease progression and doesn't bring cure. Still, the company's current valuation is about $1.4 billion. Our therapy is expected to bring cure, along with high therapeutic index and potential for sensitization to conventional therapies. We aim to submit IND in 2 years and treat our first GBM patient within 3 years, with the vision to bring to patients effective, non-invasively delivered, non-side effects bringing therapy. Our therapy addresses main pitfalls of previous p53-based therapies. Instead of p53 encoding gene delivery using viral vectors, which are highly immunogenic and have low efficacy, we use exosomes, which are non-toxic, non-immunogenic, and allow for flexible administration routes. Instead of delivering human p53 protein, which aggregates with mutant protein present in malignant cells and leads to dominant negative inhibition of wild-type p53 function, we utilize animal origin p53, which has different C-terminal domain, and this prevents aggregation and ensures restoration of the p53 function. By targeting p53, our therapy has potential to address 60% of cancers, and it is expected, again, to be synergetic with existing oncology drugs. We have performed multiple experiments in vitro showing p53 mutation-specific activity in multiple cell lines for GBM and across multiple cancer types, such as melanoma, lung cancer, and colon cancer. Our results indirectly support p53 involvement hypothesis. Watch what happens to cancer cells after adding a single drop of our therapeutic, with the field on the left almost clearing over the period of 3 days, as opposed to the control on the right, where cells continue to grow and cover the plate completely. This process dynamic is consistent with apoptosis against a cytotoxic effect. We actually observe residual viability of about 30% after 74 hours. In our survival studies with immunocompetent mice in a colon cancer models, we have shown significantly prolonged survival, reduced metastases development with no observed adverse effects. These results are statistically significant. In a recent preclinical study, we monitored tumor volume, again with immunocompetent mice in a colon cancer model, showing tumor growth arrest in the treatment group as opposed to high growth of the tumor in the control group after just 2 injections of our therapy. The treatment showed to be very well tolerated by the animals. Obviously, these results are statistically significant for the tumor size, and they were accompanied by significant histopathological findings showing reduced angiogenesis, increased apoptosis, and reduced proliferation in the treated group. Well, in the very densely populated field of cancer research, our therapy is strongly positioned at the intersection of major therapeutic directions: p53, exosome delivery, and tissue-agnostic therapy. Our road to market is aligned with major regulatory milestones and is aimed at early consultation with the FDA and continuous development under oversight of the FDA for fastest market access. We have recently expanded our facilities and are now headquartered in North Israel in one of the major industrial parks. We are currently working on raising our next round of funding and are pursuing preclinical development, production optimization and scaling, and strategic partnerships. Our intellectual property is fully owned by the company. We own 2 patent families, one of which owns oncology therapeutic and is now in national phase in multiple locations around the world. Manufacturing is kind of a holy grail of exosome-based therapies. ExoProther engaged with one of the best established CDMOs located in the US with great production, development, and manufacturing capabilities and facilities, and experience with almost 500 biologic projects, including those based exosomes. We have recently submitted joint BIRD grant, which is currently under review. This grant is a binational Israel-US Industrial Research Foundation. Our regulatory strategy is targeting first approval in the USA. We know that our therapy is going to be regulated as a biologic drug through FDA 351 pathway. As we're targeting significant unmet need, we know that we're going to receive an orphan drug indication, and we are going to be in early contact with the FDA to coordinate development. We anticipate multiple priority designations. Our team is comprised of scientists, entrepreneurs, experts in cell biology, product development, advanced biomanufacturing, and experts, unique scientific advisors. Professor Daniel Offen, head of Translational Neuroscience Lab at Tel Aviv University, and internationally recognized researcher in the field of exosomes, and himself co-founder of several biotechnology companies. Dr. Alex Panchin, an expert with almost 50 years of experience in molecular biology and immunology, and Dr. Avi Treves, an expert in translational clinical R&D. We believe that we're uniquely positioned to win with a unique mechanism of action, broad applicability. Again, 60% of human cancers should be relevant to our therapy. Synergy with existing oncology therapies, IP fully owned by the company, and strong competitive advantages. We have achieved solid, consistent preclinical results and have shown proven achievement with low burn rate. Our regulatory strategy is focused on accelerated market access, and we have potential for a high multiple exit within 5 years. Thank you for the opportunity to present at the World Stem Cell Summit. Looking forward to receiving your questions.
Thank you, Dr. Volokh. That was a very interesting presentation on what sounds like some potentially groundbreaking therapies. One of the questions I have is on how this is different from other drugs that are in clinical trials now, and just to compare and contrast what you're doing with some of the major approaches to cancer.
Thank you for that question. So first of all, indeed, there are similarities with one of the with certain approaches that are being developed in oncology therapies now, and this is pursuing a specific biomarker rather than tissue of origin. So following the GBM indication, which we're pursuing right now, we're going to expand and position this therapeutic as a tissue agnostic therapy. This is probably one of the similarities. In the sense of what makes us different is, first of all, breadth of application. As multiple times mentioned throughout the presentation, it's about 60% of human cancers that are expected to be benefiting from our therapy. Moreover, cancers associated with P53 mutation are known to be the most aggressive and also resistant to conventional therapies. So we're taking up for very important challenge.
Yes, that was a very good point that you made, that this is a mechanism in cells that leads to cancer and not specific to one tumor type. So it could be applied to the 60% of the cancer cases. One of the things that you mentioned was where you are in the development process. Could you just repeat that and talk about the milestones?
Of course. So the company is in preclinical stage, and we're expanding on our preclinical activities. Our GBM pilot study is currently in progress.
In terms of the adverse events and the side effects that one could expect from a cancer therapy, how does your therapy, what would be expected from patients when they receive the therapy?
Thank you for that question. That's actually a very important feature of our therapy. It's not cytotoxic, and delivery through exosomes is known to be non-immunogenic. So what we have seen currently so far with animals is that therapy is very well tolerated and no adverse effects have been observed so far. The treated animals had actually very good quality of life. And those of them who survived the studies and continued to live their normal lives. And this is what we expect to be happening with our human patients eventually.
That's the toxicities and side effects are an important part. So that's great to hear. So when do you expect the products to reach clinical trials and start treating patients?
We expect to treat our first GBM patient within 3 years.
Okay, and there's so much there and so much potential of using P53 in so many different tumor types. What is your strategy to go forward and develop this technology platform into all of the things that it has potential to do and reach its maximum potential?
So we're strategizing our development into 2 phases. The first being targeted for GBM indication, and that's for very important reasons. First, it's a huge unmet need, and current survival rate is so low. So basically, there is no treatment for those patients, and the clinicians are very accepting of trying new GBM therapies. In addition, it allows us to pursue orphan drug designation and multiple potential priority designations. So that would be the first stage. The second phase for us would be expanding through umbrella trials into tissue agnostic therapy, so that access to this therapy would be driven by biomarker, the P53 mutation, rather than the tissue or organ of origin.
Interesting. That's a very good point about the clinical trial. And on one hand, GBM is an orphan population with very poor survival. So you'd be able to see a signal of activity relatively quickly. And on the other hand, an umbrella trial by the marker rather than the tumor type would also give you signal of activity and proof of concept that would be important to going forward and designing additional trials.
Thank you.
In terms of XProTher as an investment, where are you now, and what are you planning to do?
So actually, right now we're fundraising, following these very solid results with the preclinical studies. We're working our next funding round. We're planning to raise $4 million, and in parallel, we're pursuing non-dilutive funding through grant systems. So actually, we're in a very pivotal point in our development.
Thank you for joining us for this C-Suite interview presentation brought to you by ChannelCheck. Visit our YouTube channel for more interview content, as well as virtual roadshows and conference presentation replays. New content is added regularly, so subscribe below to stay up to date. Visit channelcheck.com or click the link in the description to access equity research, news, and advanced market data on this and the 6,000 other small and microcap companies listed.